Okay, so ladies and gentlemen, you are all welcome to my video or to my YouTube channel. Please, if this is the first time you are coming, please try to subscribe and join the YouTube community. So in today's video, we are going to look at the concept of Krebs cycle. Krebs cycle. Krebs cycle. So this is what we are going to look at today. For this Krebs cycle is also called citric acid cycle. So we can either call it Krebs cycle or citric acid cycle. So you can either call it Krebs cycle or citric acid cycle. And this cycle was actually discovered by a scientist called Hans Krebs. Hans Krebs is the name of the scientist that actually discovered or that demonstrates the citric acid cycle. So the name of the cycle is named after the scientist. And this citric acid cycle is a series of enzymatic reactions that take place in the mitochondrial matrix, which is steps or series of reactions that take place in the mitochondrial matrix. And you should know that, ladies and gentlemen, most of these metabolic pathways, they are compartmentalized. So when we say they are compartmentalized, it means that there are organs or there are organelles of the cells where most of these metabolic pathways take place. So this metabolic pathway, which is Krebs cycles, is found only, or it takes place only in the mitochondrial matrix. So meaning that any organism that doesn't have mitochondrial matrix, it shall not undergo this series of reaction, meaning that it cannot undergo citric acid cycle. So what are the cells or organisms that doesn't have mitochondria? Like prokaryotic organisms, there are organisms that doesn't have nucleus and membrane bound organelles. So since they doesn't have mitochondria, it means that they cannot undergo citric acid cycle. In addition, red blood cell doesn't have mitochondria. So that is why it cannot also undergo citric acid cycle. In addition, we have rod and cone cells. Rod and cone cells of the eyes, they doesn't also have mitochondria. So they cannot undergo citric acid cycle. In addition, like any organism that doesn't have well-developed mitochondria, so they cannot also undergo the citric acid cycle. Now, let me just give example, like in the case of the citric acid cycle. So in the case of the cancer cell. So, like in the case of cancer cell doesn't have well-developed mitochondria, so they cannot also undergo citric acid cycle. Ladies and gentlemen, very importantly, citric acid cycle or Krebs cycle takes place in the mitochondrial matrix. It takes place in the mitochondrial matrix. So any organism that doesn't have well-developed mitochondrial matrix, it means that it cannot undergo citric acid cycle. So then, very importantly, what do we generate at the end of citric acid cycle? So a citric acid cycle, as I said, is a series of reactions where acetyl-CoA, is converted to generate NADH plus ATP plus CO2 plus water molecule plus FADH2. FADH. NADH2. So at the end of the citric acid cycle, we expect that the acetyl-CoA oxidize, it oxidizes to generate major products, and these major products include NADH, ATP are generated, CO2 is also generated, water molecules are also generated, and then FADH is also generated. But very importantly, ladies and gentlemen, in the citric acid cycle that usually takes place is utilized, or it's usually one molecule of glucose, because we need to understand that. Because remember that after glycolysis, or for each molecule of glucose, of glucose, after glycolysis, two molecules of pyruvate are generated, and of course the two molecules of pyruvate will undergo oxidative decarboxylation to produce two molecules of acetyl-CoA. So now, if you want to know exactly how many NADH, ATP, carbon dioxide, and FADH are generated, then it means that for each citric acid, for each molecule of glucose, we expect two citric acid cycles to take place. We expect two citric acid cycles to take place because for one citric acid cycle, you need one molecule of acetyl-CoA, and for two citric acid cycles, so you need two molecules of acetyl-CoA. And of course, for each glucose molecules, you generate two molecules of acetyl-CoA. So now the question is, for two molecules of acetyl-CoA, how many NADH are generated? So for two molecules, when two molecules of acetyl-CoA is fully oxidized, four molecules of carbon dioxide are generated, and then 6 molecules of NADH are also generated, plus 2 molecules of FADH2 plus 2 molecules of ATP. So you see, for each molecules of glucose, for one molecule of glucose, 2 acetyl-CoA are generated. So, ladies and gentlemen, you should understand that in cellular respiration, cellular respiration is actually a series of reactions where cellular respiration is the catabolic reactions where it's a series of catabolic reactions that take place in the cell. And this cellular respiration involves 4 major steps. So cellular respiration is involved 4 major steps. And what is this cellular respiration? It's a catabolic reaction that takes place in the cell. So usually these catabolic reactions or this cellular respiration take place in 4 stages. The first stage is actually the first step is where the glucose is converted into pyruvates, and the process is called glycolysis. So glycolysis is the first step of cellular respiration. And then the second step of cellular respiration, of course, is the conversion. So the second step of this cellular respiration is the conversion of pyruvate to acetyl-CoA using pyruvates, dehydrogenase enzyme complex. So you see, as I said, cellular respiration involves 4 steps. So the first step is actually the conversion of glucose to pyruvates. That is the first step through glycolysis. And then the second step is the conversion of pyruvates to 2 molecules of acetyl-CoA using pyruvates dehydrogenase complex. And then the third step, which is number 3, is the Krebs cycle. The third step is the Krebs cycle. Is the Krebs cycle. Krebs cycle. So the third step is the Krebs cycle, where the acetyl CoA will be oxidized to generate 4 molecules of carbon dioxide, 6 molecules of NADH, 2 molecules of FADH, and then 2 molecules of 2 molecules of ATP. So you see, this is the 3 steps of cellular respiration. So the first step is conversion of glucose to pyruvate, and then the second one, conversion of pyruvate to acetyl CoA, and then the third step is the conversion of acetyl CoA to significant equivalent, which is NADH and FADH. And then the fourth stage of this cellular respiration is when we have FADH and NADH undergo what we call electron transport chain. So the NADH2 and FADH2 do what we call electron transport chain, electron transport chain, where 6 molecules of NADH and 2 molecules of FADH will now actually undergo reduction of oxygen to generate water molecules plus ATP. So, ladies and gentlemen, these are the 4 steps of cellular respiration, where glucose is converted into pyruvate as the first step, then the pyruvate converted into 2 molecules of acetyl CoA, then pyruvate the hydrogenous enzymes complex, that is the second step, and then the third step is the Krebs cycles, where 2 molecules of acetyl CoA will undergo oxidation to generate 6 molecules of NADH, and then 2 molecules of FADH, and then these 6 molecules of NADH and 2 molecules of FADH will now enter electron transport chain to reduce oxygen molecules to water and generate ATP. So the major aim of cellular respiration is to generate ATP. So that is why generally 1 molecule of glucose in the body, 1 molecule of glucose in the body can produce about 36 to 38 ATP. So this is what actually happens. So for each molecule of glucose, there is a likelihood when it undergo the complete cellular respiration. The complete cellular respiration is going to produce either 36 or 38 molecules of ATP. And this is what we call citric acid cycle. This is cellular respiration, where 1 molecule of glucose will be converted to produce 38 molecules of ATP or 36 molecules of ATP. So what are we going to start with? First, we are going to look at the citric acid cycle profile. So the first thing that we are now going to ask ourselves is that we said that this citric acid cycle is located in mitochondrial matrix of the cell. So now the question is, how these mitochondrial matrix come into play in Krebs cycles? So first, remember, the first thing that happens during this cellular respiration is the conversion of glucose to pyruvates. And this conversion of glucose to pyruvate is all taking place in the cytosol of the cell. In the cytosol of the cell. So now the question is, how is this pyruvate gets into the mitochondrial matrix for these Krebs cycles to start? So it means that there must be a transport of these pyruvates to the mitochondrial matrix from the cytoplasm or from the cytosol of the cell. So that is why at this point we are going to look at the transport of pyruvates from cytosol to mitochondrial matrix for these for the Krebs cycles to take place. So that is the next thing that we are going to look at. So let's say here, this is where we have our mitochondria. Let's say this is where we have our mitochondria, like this. This is where we have our mitochondria. Let's say this is the matrix of the mitochondria. This is the mitochondrial matrix. So now the question is, how is this pyruvate gets into the matrix of the mitochondria? So we need to understand that for this pyruvate to get into the matrix of the mitochondria, it involves one, the origin of the pyruvate is at the cytosol. So how would it get to the matrix of the mitochondria? So there are series of steps. So first, the pyruvate can enter the mitochondrial membrane space. So the pyruvate can get into the pyruvates can get into inner mitochondrial and get into mitochondrial in mitochondrial intermembrane space. Mitochondrial intermembrane space. So the pyruvates can get into the inner or the mitochondrial intermembrane space through a process or using non-selective process. So it gets into the inner membrane space. So how is this pyruvate gets into the inner membrane space? So actually the pyruvate is a larger molecule. It is actually a larger molecule. So therefore it uses a voltage dependent anion channel. It uses voltage dependent anion channel. So the pyruvate is actually a larger molecule, so it actually gets into the inner membrane space, that is the mitochondrial inner membrane space, using a non-selective voltage-dependent anion channel. So it means that the pyruvate from the cytosol get itself into the mitochondrial inner membrane space using a voltage-dependent anion channel. So that is how it gets itself into the inner membrane space using the process called voltage-dependent anion channel. And then the next thing, this pyruvate from the mitochondrial inner membrane space, then it's now use what we call the mitochondrial protein complex, which is actually located at the membrane of the mitochondrial matrix. So that is what we call mitochondrial protein complex. So the pyruvate from the inner mitochondrial space gets into or move from the mitochondrial inner membrane space to the matrix of the mitochondria using a mitochondrial protein complex. So the pyruvates from the mitochondrial inner membrane space, they get into the matrix of the mitochondria using mitochondrial protein complex. So the mitochondrial protein complex is a complex which the pyruvate use to move from the mitochondrial inner membrane space to the matrix of the mitochondria. So this is actually what happened. And of course, we need to know, because we know that in cell, there are ways in which the organelles of the cell, we have what we call a transport across the membrane of the cell, or transport across the cell. So we have different types of transportation across the cell. So now the question is, which types of transport this pyruvate uses to get itself into the mitochondrial matrix? So ladies and gentlemen, let me repeat myself for better clarity. First, glycolysis takes place in the cytosol, and the end product of glycolysis is pyruvate. So for the Krebs cycle to take place, it requires the pyruvate to be relocated, or to relocate itself from the cytosol to mitochondrial matrix. So how is this pyruvate take itself from the cytosol to mitochondrial matrix? So first, this pyruvate can get itself to inner membrane or intermembrane space, that is the mitochondrial intermembrane space in the mitochondria, using voltage-dependent anion channel. Voltage-dependent anion channel is a type of channel, or is a channel that used to transport large molecule, and it is a non-selective method. So therefore, pyruvate is a larger molecules. So it uses the voltage-dependent anion channel to get itself into mitochondrial intermembrane space, and then for the pyruvate to translocate itself from the mitochondrial intermembrane space, it uses mitochondrial protein complex to transport itself from the mitochondrial inner membrane space to the mitochondrial matrix. So now we have the pyruvates in the mitochondrial matrix. What happened next? So now the pyruvate, we see now how the pyruvate translocates itself from the cytoplasm, or from the cytosol, to mitochondria. So now the next thing is, what happened to the pyruvate? Okay, so the pyruvates in the mitochondria, because this is also one of the steps of cellular respiration, where we are going to convert, where the pyruvate will convert to acetyl-CoA. So the pyruvate is converted to acetyl-CoA using an enzyme called pyruvate dehydrogenase complex. That is the name of the enzyme that convert acetyl-CoA. So pyruvate is converted to acetyl-CoA using an enzyme called pyruvate dehydrogenase complex. And that is the name of the enzymes that convert the pyruvate to acetyl-CoA. And whenever you have the word complex, it means that it is a complex, it is not actually a single enzyme. It is a complex enzyme that encompasses so many other enzymes. So a pyruvate dehydrogenase complex is a complex of three enzymes. It's a complex of three enzymes. We have E1, we have E2, and then we have E3. So these are the three different enzymes that are encompasses this pyruvate dehydrogenase complex. It means it is an enzyme that encompasses so many other enzymes. So pyruvate dehydrogenase enzyme complex, it comprises of three enzymes. So the E1, which is the first enzyme, is pyruvate dehydrogenase. The first one is pyruvate dehydrogenase. That is the first enzyme here, which is E1. And then the E2 is dihydrolycoyl transacetylate, is dihydrolycoyl transacetylase, transacetylase. So we have dihydrolycoyl transacetylase is the second enzyme. And then the third one is dihydrolycoyl dehydrogenase, is dihydrolycoyl hydrogenase. So these are the three enzymes that form the hydrogenase, or that form pyruvate dehydrogenase enzyme complex. And this reaction is a decarboxylation reaction and transacetylation reaction. This reaction involves transacetylation and decarboxylation, because pyruvate is a three-carbon compound. So when these pyruvate dehydrogenase complex enzymes act on the pyruvates, then an acetyl-CoA is generated, and acetyl-CoA is a two-carbon compound. It's a two-carbon compound, while the pyruvate is a three-carbon compound. So it means that there is decarboxylation reaction. So therefore, at the end of this reaction, we expect that one molecule of CO2 is generated. So pyruvate is converted to acetyl-CoA with the removal of CO2, and then after this, we have pyruvate converted to acetyl-CoA plus one molecule of CO2. Then after this, what next? These enzymes require a lot of cofactors. It requires a lot of cofactors and coenzymes for these enzymes to work, or for these enzymes to convert pyruvates to acetyl-CoA. And some of these cofactors, there is N, we have four enzymes A. We have coenzyme A. We have coenzyme A. We have NAD plus is also another cofactors. And then apart from NAD plus, we have TPP, that is thymidine pyrophosphates. It also requires lipoids. How many cofactors so far? Also the coenzymes, we have four different coenzymes here. And then after this, there is also FAD. There is FAD. So you see 1, 2, 3, 4, 5. So it means that for these enzymes to work, 5 coenzymes is required for the pyruvate to be converted into acetyl-CoA. And at the end, another molecules of NADH is generated. So you see, pyruvate is converted into acetyl-CoA through decarboxylation by removing CO2 with the use of enzymes pyruvate dehydrogenase complex. And this pyruvate dehydrogenase complex is an enzyme that encompasses 3 enzymes, E1, E2, and E3. And the enzymes require 5 different cofactors for each to generate the coenzyme, the acetyl-CoA. And very importantly, we should understand that at this point, when we are counting that one molecule of NADH is generated. And of course, remember in glycolysis, two molecules of pyruvate is usually generated, and then two molecules of acetyl-CoA is generated. So these enzymes or this step is also one of the major regulatory pathway in terms of metabolic regulation. So you can regulate cellular respiration at this level. You can regulate cellular respiration at this level. Like for example, this pyruvate dehydrogenase enzymes complex can be inhibited when you have the following, so when you have an increase in the ratio of the following. When you have ATP, ADP, ATP, ADP ratio is high, or when you have NADH, NADH ratio NAD plus, or acetyl CoA, acetyl CoA ratio for A. So when the concentration of the following ratio goes very high, they inhibits the pyruvate dehydrogenase complex enzyme. They inhibits pyruvate dehydrogenase complex. So what does that mean? This is actually the first step of regulating the pyruvate dehydrogenase complex enzyme. So it means that from here, when we say ATP to ADP ratio is high, that indicates that the amount of ATP is very high in the cell, indicating that there is no need for Krebs cycle, because the major aim of Krebs cycle and electron transport change is to generate energy. This point, if the ATP is very high, it is an indication that the cell has amount, or the cells have amount of ATP, indicating that there is no need for further citric acid cycle and electron transport change to generate the energy. In addition, when NADH ratio to NAD plus ratio is high, it is also another indication that means that indicate that there is enough NADH plus, because the major aim of the Krebs cycle is to generate NADH for the continuation of, for it to continue to electron transport change to generate energy. So when you have NADH slash NAD plus ratio is high, it means that NADH is very high in the cell, indicating that there is no need for electron transport change to generate more of this NADH. But when it is low, and this one also when it is low, it is also an indication that there is low ATP in the body when this ratio is low. And when this ratio is low, it means that there is low NADH in the body, indicating that there is a need for the continuation of citric acid cycle. And then acetyl-CoA, the major work of this enzyme to generate a lot of acetyl-CoA for the continuation of Krebs cycle. So if you now have an acetyl-CoA ratio for A to be very high, it means that there is enough of acetyl-CoA in a cell, meaning that this enzyme is not longer needed to be activated to generate more of the acetyl-CoA. So therefore, it will be inhibited. So this is actually one of the states where you can control the activity of pyruvate dehydrogenase complex. So this is actually the first step. And then the second one of this metabolic regulation is that when you have, there are two enzymes that are usually activated. Sorry, two enzymes that are usually activated at this point. There is an enzyme called pyruvate dehydrogenase kinase and pyruvate dehydrogenase, pyruvate dehydrogenase kinase, and pyruvate dehydrogenase phosphatase. Phosphatase. So this is also some of another, or these are also key enzymes that are involved in activation, and that is the covalent activation, because in metabolic regulation we have covalent modification of enzymes activity. So you can control enzymes using covalent modification. So when we say covalent modification, it means by adding a particular group. So when we say kinase, the word kinase means is an enzyme that transfers a group to an existing compound or molecules. So at this point, if we have, for example, this enzyme is activated, that is the pyruvate dehydrogenase kinase, is an enzyme that actually involved in phosphorylation. Phosphorylation. When we say phosphorylation, it means addition of phosphate group. Phosphorylation means addition of phosphate group. So usually in the pyruvate dehydrogenase, when the enzymes, which is pyruvate dehydrogenase, is phosphorylated, so usually this phosphorylation is usually taking place in the serine residues, three serine residues of the enzyme. So when you say three serine residues, it means that in the active site, generally these enzymes work by binding with the enzymes at their active site. So when you have a substrate, let's say this is your enzyme. So this is the enzyme, and then this is the active site of the enzyme. The active sites of the enzyme, that is where the substrate binds. So usually at the active site of the enzymes, there is what we call amino acid residue. So it means that at this pyruvate dehydrogenase complex enzymes, they have serine. They have three serine. So generally serine, they have OH2. So what happened is that this pyruvate dehydrogenase kinase enzymes, it's usually what it does, it phosphorylates these three serine residues. So it means that it adds phosphate group to these three different serine residues at the active site. So once they are phosphorylated, then the enzyme should become inactive. So it means that you can control this pathway by activating or by increasing the activity of pyruvate dehydrogenase kinase enzyme. So what it does, it phosphorylates three serine residues, making the pyruvate dehydrogenase complex enzymes to become inactive. Then what is the work of the pyruvate dehydrogenase phosphatase? It gives actually the reverse of pyruvate dehydrogenase kinase. So kinase adds phosphate, and then phosphatase remove phosphate group. So once these phosphate groups are removed using the pyruvate dehydrogenase phosphatase, then the enzymes will now become active. So this is actually equivalent modification of activating and inactivating enzymes using pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. So then there is also the next one is also allosteric regulation. So which of the enzymes that are usually activated or inactivated by pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase? Because we said that the complex encompasses of three enzymes. Is it that the phosphorylation and dephosphorylation is for all these three enzymes? E1, E2, E3, or only one of the enzymes? It's actually on only E1, because it is the master enzymes here. So it means that if you are able to phosphorylate or dephosphorylate the E1 enzyme, that is pyruvate dehydrogenase enzymes, using the kinase phosphatase, it means that you can control the enzyme. So phosphorylation or dephosphorylation can serve as a mechanism of controlling or regulating the activity of the pyruvate dehydrogenase complex specifically E1, so that it can either use these regulatory mechanisms, either operate, that is, increase the formation of acetyl CoA or decrease it, or you can even stop it or off it. So you can on it, you can off it, you can upregulate it and downregulate it using these pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase, specifically acting on E1 enzyme. So then the next thing is also we have what we call allosteric inhibition, that is the third regulatory steps. So the third regulatory step is allosteric regulation, is allosteric, allosteric, allosteric inhibition. So you can call it just allosteric inhibition. So generally enzymes, apart from their active sites, they have what we call allosteric sites. We have what is called allosteric site. So allosteric site is another site apart from the enzyme active site. So a molecules can bind to this allosteric site, and once a molecules bind to this allosteric site, it usually, or in most cases, it affects the active site of the enzyme. So allosteric inhibition, it may be the end products of the pyruvate dehydrogenase enzymes that can actually serve as an allosteric molecule. Like for example, the product of this enzyme pyruvate dehydrogenase complex is acetyl CoA. So when there is enough acetyl CoA in a cell, it can serve as an allosteric modulator, where it can bind to the allosteric site of the enzymes and inhibit its activity. So allosteric site is a site on an enzyme where the end products, or where the molecules can bind to it. That molecules can be end products of that enzymes or another molecules. So that is allosteric inhibition. So this is actually three major mechanisms in which the activity of pyruvate dehydrogenase enzymes can be regulated. So then, so ladies and gentlemen, first we started with glycolysis produce pyruvate, pyruvate produce acetyl CoA, and then the acetyl CoA will now continue with with citric acid cycle. So very importantly, the pyruvate is generated in the cytosol, and it's also transported to the mitochondrial matrix using what we call protein co-transport. Although I didn't mention that during transportation of pyruvates from cytosol to matrix, mitochondrial matrix. So what types of transports does it use? Because as I said, we have different types of transport. We have passive transport, we have active transport, we have facilitated diffusion, and we even have simple diffusion. So actually the type of the transport used by these pyruvates to the mitochondrial matrix is actually, we call it protein co-transport. We call it protein co-transport. Sorry, proton co-transport. Proton, we call it proton co-transport. So remember, ladies and gentlemen, one of the transport systems across a cell, we have what we call co-transport. It can be symport, it can be antiport. So when two molecules are moving in the same direction across a cell, it is called symport. And when it is following in opposite direction, like for example, if one is following in and the other is going out, and that is called antiport. So it is proton co-transport. We call this transport of pyruvate to mitochondrial matrix as the proton co-transport, is because the proton and the pyruvate move in the same direction from the cytosol to the matrix, from the cytosol to the matrix of the mitochondria. So it means that this proton co-transport of pyruvates and protons from the cytosol to mitochondrial matrix is called symport. It is a symport transport. So now, ladies and gentlemen, we are now going to look at citric acid cycle in details. So, ladies and gentlemen, this is citric acid cycle and it is very simple to explain. So first, we have talked about the formation of acetyl CoA. So when acetyl CoA is generated, so in the mitochondrial matrix, this acetyl CoA and oxaloacetate condense themselves. So they will condense. When they condense, means that they will combine using an enzyme called straight synthase, then it will produce what we call straits. These straits can undergo hydration. Hydration, when we say hydration, means addition of water molecules to generate what we call cis-aconidates. Cis-aconidates. So these aconitase enzymes through hydration reaction, although it is actually a reversible reaction, so using an enzyme called aconitase, it will produce cis-aconidates. The cis-aconidates will further be converted into isostrate using an enzyme called still the same enzymes aconitase. So aconitase will convert cis-aconidates to isostrates, and then this isostrate will be converted to alpha-ketoglutarate, which will be converted to alpha-ketoglutarate by the work of an enzyme called isostrate dehydrogenase. So isostrate dehydrogenase is an enzyme that converts isostrate to alpha-ketoglutarate. And this is one of the steps, as you can see clearly, at this step, one molecules of NAD plus. So at this stage, one molecules of NAD plus is utilized. That is used from here. NAD plus is utilized to produce one molecules of NADH. So it means that one of the steps, it is actually a reversible, so it's an irreversible step where isostrate dehydrogenase convert isostrates to alpha-ketoglutarate. So one molecule of NAD plus is utilized to generate one molecule of NADH. So it means that one molecule of NADH is generated. And then the next one is the conversion of alpha-ketoglutarate to succinyl-CoA. So an enzyme alpha-ketoglutarate dehydrogenase, alpha-ketoglutarate dehydrogenase enzymes complex convert alpha-ketoglutarate to succinyl-CoA. And at this stage, also another molecules of NADH is utilized. So this step also take one molecule of NAD plus. So one molecule of NAD plus also utilizes alpha-ketoglutarate dehydrogenase to generate another molecules of NADH. Then the next step is, of course, the conversion of alpha-ketoglutarate to succinyl-CoA by the enzymes alpha-ketoglutarate. So the next is actually when we have the succinyl-CoA, which will convert to succinate. So at this step, succinyl-CoA synthase is an enzyme that utilizes. So you can see clearly that this step we utilize one molecules of NADH. Sorry, one molecules of GTP is GDP. So sorry, GDP. So GDP, one molecule of GDP is utilized, and then one molecules of GTP is generated. So GTP is the same thing as ATP. So it means that this is one of the steps of the succinyl-CoA where a molecules of ATP is generated. So GTP is the same thing as ATP. So it means that this is one of the steps that the ATP is generated. So now, this is one of the steps that GTP or ATP is generated, and this is what we call a substrate level phosphorylation. Remember, in one of my videos, I told you that there are two ways in which energy is generated. One, we have electron transport chain, and second, we have substrate level phosphorylation. So when we say substrate level phosphorylation, it means that it's a state in which ATP is generated from the substrate. ATP is generated from substrate. Like for example, at this stage, the inorganic phosphates plus GTP using an enzyme succinyl-CoA synthase generates GTP. So it means that this energy is extracted or is produced from succinyl-CoA. And that is why it is called substrate level phosphorylation, because the succinyl-CoA is a substrate for succinyl-CoA synthase. So we generate ATP from substrate. Then we have succinate. So the succinate, using an enzyme called succinate dehydrogenase, will be converted to produce fumarate. And at this stage, the succinate dehydrogenase enzymes produce one molecule of FADH2. It produces one molecule of FADH2. So then the fumarates will be converted to malate using fumarase enzymes. So fumarates will be converted to malate fumarase enzymes, and at this stage, the malate that is generated is converted to oxaloacetate using malate dehydrogenase, and this is also one of the steps where NADH is also generated. So you can see clearly 3 molecules of NADH are generated, 1 molecule of FADH2 are generated, and then 1 molecule of ATP are generated during citric acid cycle. So now, ladies and gentlemen, very importantly, we say that CO2 is also generated during electron transfer from our introduction. So at what step at what steps these molecules of CO2 is generated? So the first one is actually the conversion of isostrates to alpha-ketoglutarate. The conversion of isostrates to alpha-ketoglutarate at this stage, 1 molecule of CO2 is actually generated. So it means that this is actually a decarboxylation step where 1 molecule of CO2 is generated. So there is a decarboxylation at this stage. And then the next step is actually conversion of the alpha-ketoglutarate to succinyl-CoA. So the conversion of alpha-ketoglutarate to succinyl-CoA at this stage also another molecule of CO2 is generated. So you can see clearly that 2 molecules of CO2 is generated. So you see we have isostrate dehydrogenase at this stage, CO2 is generated, and 1 NADH is generated. Also, at the stage where we have alpha-ketoglutarate dehydrogenase complex, 1 molecule of CO2 is also generated. So, ladies and gentlemen, we can see clearly from 1 molecule of acetyl-CoA, we are able to generate. You see, at least we have 1 CO2, 1 CO2, then we have 2 molecules of CO2 plus 3 molecules of NADH2. NADH plus there is 1 FADH2 plus 1 FADH2. Then plus apart from the FADH2, there is also ATP or GTP, so plus 1 molecules of ATP. So this means that for the citric acid cycle to occur, first you need acetyl-CoA, you need, apart from acetyl-CoA, you need water molecules. You see, there is hydration, so 1 molecule of water is needed. And then there are 3 molecules of NAD plus 1 at this stage, 1 also at this stage, and also another 1 at this stage, NAD plus. And then we have also plus 1 molecule of FADH. So here also FAD plus, FAD plus also into the cycle. So we have plus FAD plus FAD plus then plus also GDP or ADP, sorry, plus ADP or GDP is the summation, then plus inorganic phosphate. So you see, what you need here is you need acetyl-CoA plus water molecule plus 3 NAD plus 1 FADH plus 1 ADP plus inorganic phosphates to generate this product, and this citric acid cycle is the 8 steps reaction. It is 8 steps reaction. So ladies and gentlemen, I think without wasting much of our time, it is very clear. So this is how electron transport chain take place this is how citric acid cycle take place in our body. I hope this video is very clear and you can all clearly see how it started from pyruvate, how the pyruvate is transported from the cytoplasm to mitochondrial matrix. Then from the mitochondrial matrix, the pyruvate dehydrogenase enzymes complex converts the pyruvate to acetyl-CoA, and then the acetyl-CoA here enter the 8 series of reaction steps where 2 molecules of water, 3 molecules of NADH2, 1 molecules of FADH2, and 1 molecules of ATP. And remember that I told you that for 1 molecules of glucose, it is usually 2 acetyl-CoA that is generated. So it means that at the end of one complete oxidation of glucose, we are going to have 2 citric acid cycles. So if it is 2 citric acid cycles, we are going to multiply everything here by 2. So ladies and gentlemen, don't forget to join and subscribe the YouTube community. Thank you very much and see you in my next video.